This is Group 12 video presentation. Did you know that there are 2.2 billion people who are visually impaired and around 39 million people who are completely blind worldwide? Being visually impaired impacts people's day-to-day life, and there used to be no cure. Scientists try to cure a type of childhood blindness called Leber congenital amaurosis type 2 through gene therapy. This type of childhood blindness is caused by mutation in genes that code for a protein, RPE65, that participates in chemical reactions that are needed to detect light. A treatment method was being developed simultaneously by groups at the University of Pennsylvania and at the University College London and Moorfields Eye Hospital.
In the treatment, scientists inserted a healthy copy of the mutated gene directly into the space between the retina and the pigmented retinal tissue where the chemical reactions take place. This gene helped the retinal pigmented epithelium cell produce the missing protein that is dysfunctional in patients. Although the patient's visions did improve, it is not a permanent fix. For the first 1 to 3 years, the patient's vision improvement peaked, but after that, they increased visual sensitivity and kept losing photoreceptors, the cones and rods responsible for daytime and nighttime and darkness vision.
The scientists needed something new. In comes CRISPR. The eye is the ideal organ for testing new therapeutic approaches, including CRISPR, because it is easily accessible and retinal tissue in the eye is shielded from the body's defense mechanism, so the body will not launch a defense attack on it. Scientists started working on a way to use CRISPR to cure blindness. The scientists at Adidas Medicine first showed safety and proof that the CRISPR strategy worked in cells extracted from patient skin biopsy and in non-human primate animals. These studies led to the formulation of the first ever human CRISPR gene therapeutic clinical trial.
The patients receive a dose of the therapy while under anesthesia when the retina surgeon uses a scope, needle, and syringe to inject the CRISPR enzyme and nucleic acids into the back of the eye near the photoreceptors. To make sure that the experiment is working and safe for patients, the clinical trial has recruited people with late-stage disease and no hope of recovering their vision. The doctors are also injecting the CRISPR editing tools into only one eye. Gene therapy that involves CRISPR promises a permanent fix and a significantly reduced recovery period. A downside of the CRISPR approach is the possibility of an off-target effect in which another region of the cell's DNA is edited, which could cause undesirable side effects such as cancer. However, new and improved strategies have made the likelihood of this exceptionally low.